Hi, hello, and welcome to this final presentation. I'm Uroš Smajdek and I come from University of Ljubljana, and today I'll be talking about molecular visualization of biological specimens in virtual reality using Foveated RTX ray tracing. So this topic was motivated by a general rise in interest in the molecular assemblies and general features of different viruses during the pandemic. For an example, this COVID model has sort of become an icon of those times, and also terms such as spike protein, membrane, RNA have really entered the public's vocabulary during the sad times where we're closed in. Also, now we have new and better ways to construct such models. For an example, this is a paper from 2020 where they showcase a rapid way to construct scientifically accurate models from just a few two-dimensional microscopy scans. Such visualizations can then be used to disseminate the scientific information to broader public. They can aid in scientific presentations, and they can also be used for scientists themselves to glean new information. And also all of these aspects are further enhanced by usage of AR and VR technology. Now, over the years, there have been really a lot of different ways to visualize molecular data. I've just gathered a small sample of them, but most importantly, the final one by Harvey et al. In a still unpublished paper, the authors present a way to visualize 5 trillion atoms in real time on a latest GPU using RTS ray tracing and on-demand scene generation. Although the mentioned method is really designed for desktop and rendering on a full HD monitor, but we are working with virtual reality headsets. Specifically, this was done on MetaQuest Pro, which has a per eye resolution of about 4 million pixels, which in total is 4 times as much as a full HD monitor. Additionally, rendering has to be done twice for each eye separately. And on the topic of performance, we really should not go below 60 frames per second, as it can induce headaches on the user, and the headset itself recommends 72 frames per second. So to bridge these two aspects, we use the previously mentioned foveated rendering together with RTX ray tracing. So quickly, what is ray tracing? It's a rather conceptually simple algorithm that was already thought of in 1968, although that is today known as ray casting. Essentially, the algorithm describes a process where we shoot rays from the camera through the final image and into the scene until a ray hits an object. We can then use the properties of the hit object to compute the final color of the pixel. Later on, there were further additions, and this is now today referred to as ray tracing, where we can shoot ray from the hit position towards the light source. That way we obtain shadows. We can also trace the ray further in the scene, simulating light reflections, thus obtaining global illumination. Although we, in this case, do not use any of those additions, as one, they are an incredible drain on performance, and secondly, we are really working with an abstract environment that doesn't really have any physical materials, and it would not really provide additional information to the user. Now, the most complex part of this operation is heat detection itself, as technically speaking, for each ray we have to check the collision with each object in the scene, and we are working with millions of objects. Now, this is where RTX ray tracing comes in. Beside offering simplified scene generation, more importantly, it offers hardware-accelerated heat detection, where it takes the RT cores present on the hardware, the modern Nvidia GPUs, to quickly compute the hits with all the objects in the scene and effectively abstracts this part of the algorithm. Now, what's left to the programmer is basically to define a ray generation algorithm and what happens on certain events, like when the closest object is hit or when none of the objects are hit in the scene. Maybe more interestingly, foveated rendering is a principle that basically tells us that we can focus our rendering performance in the direction of the user's gaze. The term comes from fovea, which is the part of the eye that is only responsible for the central 2% of our vision, but takes up to 50% of our visual cortex. essentially meaning we perceive detail really well where we are looking, and in the periphery we mostly perceive movement. Taking from that, we can render in higher quality where the user is looking, and that we can adjust this dynamically. This is today possible because modern VR headsets have eye tracking, and we also believe that more and more headsets will have such technology in the future. Now, to bridge these two concepts, we use something we dubbed visual acuity test. For this, we essentially split the final result discretely into three different parts with lower resolution in the periphery. And during ray generation step, we determine the number of rays cast depending on the eccentric angle from the center of the user's gaze, thus generating such image. This is also nice with ray tracing, as it goes like really well together with such methods with no overhead as opposed to rasterization. Perhaps something else that's interesting, we had to deal with two different outlooks you can have on such a molecular model. There is a macro outlook where you're interested how the different structures relate to each other. For example, how the RNA interacts with neighboring structures. So for that reason, we colored same instance of a structure with the same color, so we get all the RNAs colored in white and so on. That way we can really see the contrast between the different types of structures. But on the other hand, when you get to micro level, the composition of the individual structure, such coloring is no longer useful. So we simply interpolate the color as you get closer to a wholly different color palette, which lets the user inspect the structure itself up close. Something that we didn't expect would be needed as much is ambient occlusion. As early on we noticed this general lack of depth, lack of precision in the rendering, since we use a lot of same-shaped objects colored samely. But with ambient occlusion, the objects are much easier to distinguish, and also we have enhanced depth perception, as all of the objects are of the same shape, and user can easily infer the distance by looking at the size of the object. Finally, in the post-processing stage, we first of all had to solve a problem where, as I mentioned before, we colored the same type of structures with the same color, which made neighboring instances of the same structure indistinguishable from each other. So we add contours, like borders, around different instances of the same structure. You can also see that it makes the image, even far away, look more detailed. And from then on, we're mostly dealing with aliasing problems, because it causes flickering pixels which are also visible in the periphery, and they were incredibly distracting for the user. To that end, we blurred the image in the periphery, the lower resolution part. We used Nvidia's DLSS as temporal anti-aliasing measure. We used depth fog to hide these really far away objects that only covered a few pixels. I cannot show the anti-aliasing measures on the 2D image, so I prepared a video. This is basically me using VR headset to fly around the model. And the model itself, I can also use this. Essentially, this is a COVID-2 model submerged in blood plasma, and this was rendered with RTX 4090. I'm not sure how visible it is since we are outside, but you should see, like, what I'm looking at, the image becomes higher quality. This is just me cruising around, having some fun. And as I get closer to objects, you should see that, like, color palette changes. And I will be COVID in front of us. I'll be going back in through the membrane soon. Come on. And yeah, this is essentially the end result at this point. We also performed an evaluation. First of all, we evaluated both on desktop and a VR, in VR headset. Now, you may notice that we used increased resolution here, and this was because the headset is kept at 72 frames per second. And if we were to visualize in the basic resolution, we would go much over this frame rate in the best possible result. So we try to increase the resolution until we reach the level of until we reach the 72 frames per second and stop there. Just to mention, this whole project was developed with Vulkan graphics library and with OpenXR to bridge the gap between desktop and VR headset. Now the results show a clear improvement using foveated rendering, although very surprisingly at the time, screen space ambient occlusion algorithm that we used incredibly tanked performance, which we are not exactly sure why. It could just be that with this kind of resolution, essentially the algorithm becomes unusable. Maybe it was our implementation. We are currently investigating that. So in the future, we would like to try other ambient occlusion algorithms to solve this issue. As you might notice, we also lacked comparison with other works that I mentioned early on, and we would like to expand this to make this paper more relevant, or this method, since paper is published here. Would like to include more criteria in the visual acuity test itself, since according to some research, the perception fall-off is lower on the horizontal plane as opposed to vertical plane of our vision. And most importantly, and something that Eva mentioned in the morning, is that we would like to use this renderer as a sort of part of a larger application that would serve to give user a tour through a molecular environment, pointing out different structures, complete with voiceover, labeling, the ability of a user to select where they want to go, and so on. So this is what we are currently working on. And with this, I would conclude. I would like to thank you all very much for listening. I also have to give thanks to Ivan Viola and the whole Nanovis team at KAUST, without whom this project would not be possible at all. To this, thank you.
So, thank you. Are there any questions?
I think you spoke about aerial perspective on one slide. Like this is probably drawing the molecules in the back like under a fog.
Could you repeat that?
I think you mentioned something like aerial perspective on one slide, or I don't know if this is the phrase you used, but the idea would be like drawing the molecules in the back like covered a bit by a fog. So
oh yeah, we use depth fog, right?
Yes, yeah. Yes. But on the pictures I didn't really notice.
Yeah, because on just the pictures, I was inside the virus itself, where the depth is not high enough to actually enable the fog. But in the video, when I was cruising outside, you should have seen the red background, which is essentially the fog itself. So it was not visible on those images.
Thank you.
Hi, thank you for the presentation. The results look awesome. I had a question regarding the memory consumption, if you have any problems with that, because I noticed you stored like 74 million molecules. Yes. How much memory on the GPU did it take?
I think it took around 8 gigabytes, although we're using RTX 4090, which has 24 gigabytes, so it wasn't really a problem. It also helps that this method is really designed to be used with RTX ray tracing, which inherently requires a modern GPU.
I see. Okay, right. Thank you.
Does your SSAO get slower when you get close to some objects?
Sorry, could you...
Does your SSAO get slower when you get close to some molecules?
I'm not sure. We have.
So check that, and if yes, then we can tell you how to fix that, because I don't know which SSAO are you using, but if you're using the one from the old framework, we fixed that. But that was the issue with, when you were getting closer to molecules, it was getting slower. So if it's the same speed, if you're far away from everything, and if you're close, then that's a different issue that you have.
I implemented it like on my own, and I basically used a hemisphere version. I mean, essentially, it's an ancient algorithm by this point. But
yeah, check the thing. If you get closer to the molecules, if it's getting slower, and then if it is, then talk to Ivan so that he contacts, he puts us in contact. I will tell you how to fix that. Okay?
Okay.
Okay, I think there were some questions over here.
Yeah, I had a question that regards to the sampling. So on the borders, you essentially do sparse sampling, if I understood correctly. So in each frame, you sample just some of the pixels. Do you filter the results yourself, or you just let the LSS to filter in the temporal domain that maybe in the next frame you will sample different pixels?
We don't do any reuse of previous frames. And during a frame, we basically just render in raw resolution on the periphery and then upscale these results using the hardware upscaling, essentially.
And did you experiment with like different fall-off functions for the subsampling, which is still acceptable to the user or not?
We let the user define the actual like size of different regions because like perception differs from a person to person and also gets worse with age, so you cannot really find like the most optimal sizes that would work for everyone.
And what was the subsampling that was used at the very periphery? How many samples compared to the central part? Maybe you were mentioning 1/16. Okay.
Yeah, it was on the show and I didn't mention it.
Thank you.
Any other questions? Okay, then maybe one last question from me. If you have a user which is moving around like the eyes rapidly, does this influence the performance?
No, because we don't reuse any previous frames. We essentially render everything anew. Although if user would do that, this is not really part of the question, but even at the current headsets, there is some slight delay between the eye moving and the headset detecting eye movement, so it can feel a bit odd still.
Okay, thank you. Okay, this was the last talk for today, so now you have some free time. Definitely check out the tour to Molpir, it's awesome. And then, yeah, see you at dinner or tomorrow for the next talks.